Yeah.
Okay, you guys. Hi, my name's Ben Trenny, and we're going to learn about epizootic hemorrhagic disease, and I'm going to show you an example of what it looks like. Oh, awesome.
I mean, if you call them, if it's something beyond their
This is a deer. I'm sure that you guys all know that by now by looking at it. And so, we're going to first just go through the name of the disease. Epizootic. Epi means widespread. Zootic is a term for a virus that only infects animals. You can't get it. Humans cannot get it. And this is likely a case of EHD, or epizootic hemorrhagic disease. And hemorrhagic means there gets hemorrhages. And the reason why I think that is, a couple of reasons. This spot still has its velvet on it, which means it would have died sometime in August, early September most likely, since it's lost some of it, but it still has some of it. And also its fur color is red. The reddish fur color is a summer fur for a deer. So it likely wasn't killed by someone who shot it, because likely they would have taken the horns off it. That oftentimes happens if it was illegally harvested this early in the year. So it likely died from EHD. So we'll go through. Where can I go next? Right here. Okay, so epizootic hemorrhagic disease is a disease of a virus. It's from the genus Orbivirus. This virus is a double-stranded RNA that infects ruminant ungulates, hoofed animals with four stomachs. So this can affect wild species besides deer, such as pronghorn, bighorn sheep, mule deer. It also can infect livestock, such as cattle, goats, sheep, but EHD tends to be more common in wild game. And so epizootic hemorrhage disease is often referred to as EHD or blue tongue disease. Blue tongue disease is a little bit of a misnomer because there's a separate virus called blue tongue virus that does almost the exact same thing, except it's more deadly to livestock rather than to wild game. So it oftentimes caused huge wipeouts in Israel and a lot of the Mediterranean to cattle and sheep. And EHD is very widespread amongst wild ungulates, particularly white-tailed deer. It has to deal with white-tailed deer's habitat preferences and a life cycle of the host carrier that gives this disease. And it can impact livestock, particularly cattle. It doesn't kill cattle, but it likely will make them very sick. And then you have to give them lots and lots of antibodies to be able to cure it. So, how does the virus get into the ungulate? The virus is carried by a biting midge. You've probably seen them before. They're called sandflies or no-see-ums. They're about a quarter of the size, half the size of a mosquito. They oftentimes come in swarms. You see them when you go to a lake or like a pothole or something of that nature. They're really small. And so these midges hatch from in wetlands, and then they are little aquatic larvae, and they hatch from that. And the midge then can fly about a mile or two, and it finds a deer or some sort of ungulate. It bites it, and it carries the virus. The virus gets into the deer. And we see this appear in the summer and fall, and then the virus disappears immediately after the first killing frost. They likely believe that the virus does not make it through the winter, and it just persists in warmer climates, and then over time, over the summer, it ends up spreading all the way up into South Dakota, Montana, Canada, and so on and so forth. So what does the virus do? So once the virus gets into the deer, it takes about a week before any symptoms begin to start showing. And when the virus first takes place, the first place you notice what it will do is it will start infecting this part of the mouth right here. And it starts to get ulcers. And usually here are some sort of hemorrhages in its mouth. And it starts to get other hemorrhages elsewhere, but that's where it begins. And it starts to create excess saliva, and it develops fever. And so from this, it becomes dehydrated, and it oftentimes will go to water. During the meantime, it starts to also get a lack of oxygen in the circulatory system, which causes swelling in the head, neck, and tongue. And when it swells, because of the lack of oxygen and the swelling, its tongue becomes blue, and it hangs out of its mouth. And it can sometimes choke to death on its own tongue. But they go to the water and to cool down, and oftentimes they die near the water, like this deer here, which is where we found this deer. We were pheasant hunting last January, and we found this in the middle of a dried up slough hole. And so, why is this important that it dies near the water? The midges hatch, And the first thing they find is a dead deer that's full of blood that's been infected with EHD. They bite the dead deer, and then they can fly away, or the wind takes them somewhere between 1 to 20 kilometers away. They bite a different deer, deer gets infected. So it continues to spread. How widespread is this? This was last year, and all of the dots recognize cases that are most likely or confirmed to be EHD throughout South Dakota. And there's a few thousand last year. 2012 was a massive wipeout year. They think there's over 10,000 cases for our state alone. And it can be quite devastating. In the 80s, there was one case, not 80s, the 70s, between South Dakota, North Dakota, Montana, and Wyoming, about 80,000 deer and 4,000 pronghorn were killed. And so it can be just very much a big wipeout of these wildlife species. So, take-home points. Epizootic hemorrhagic disease is a viral disease that can kill large populations of wild ruminants, especially white-tailed deer. Epizootic is a virus that has a widespread impact on non-human organisms and cannot be transferred to humans. So, if you found the dead deer in a slough hole and you were in a survival situation, you needed to eat it, you could cook the meat and you'd be fine. You cannot get this virus. So you will not get lovely hemorrhages in your mouth and blue tongue.
From a GFP standpoint, is there anything you can do to manage it?
It's kind of hard to manage because you don't know when the virus is going to outbreak. Usually there's some environmental factors such as really hot and dry years. It tends to be worse because then you get more concentrated towards specific water areas, especially rivers, and rivers are real corridor ways. And so then you tend to see it in those years. And so you can begin to maybe do some antibody stuff. Probably the biggest management tool is they give refunds to hunters who have drawn tags for these areas so that there's less hunting pressure during that year to try and let populations bounce back. But there's not a lot that can be done because it's really hit and miss with antibodies and applying it to wild ruminants. I mean, how do you get them corralled into an area and actually apply the veterinary procedure on it? So,
And when that deer dies like near water, you know, like coyote and probably mountain lion or what too are going to come in and eat that deer. It doesn't go to them.
Yeah.
You don't think? Or
when we found this was the only part of it we found. Everything else was gone except the bones. The bones were still there, and likely were eaten away by coyotes. Coyotes are more so than mountain lions or scavengers, so you expect coyotes, and then you can even see the horns here are starting to get chewed off by mice. So mice, particularly field mice, voles, other sorts of creatures are going to come in and eat it. Also, recent finding is deer themselves are oftentimes will come and find other dead deer and begin to chew on parts of them to get some minerals and nutrients that they can't get in other sources. So,
So what is an ungulate? Is that a synonym for ruminant?
an ungulate
Is it a
is
tetra?
a hoofed animal. It's a hoofed animal. An ungulate is a hoofed animal. So that would be deer, cattle, sheep. You begin to name it. Horses aren't ungulates because they have a different sort of toe.
Nor are they ruminants.
No.
Can't you do something to the midge population?
You could begin to apply some sort of applicable spray. It's just oftentimes, let's flip back to the map here. These are pretty rural areas. And beginning to spray every single wetland becomes such an expensive policy that you're going to see. Again, you can begin to do something, because you even notice around Sioux Falls, there's less cases. Let me look right here. There's a couple of dots. But as we move more into wetter areas where there's more wetlands and less resource to begin to spray, then you'll see the same effect. On national scales, you mainly see this in the Great Plains and the Deep South, particularly Louisiana, Alabama, Mississippi.
It's wet.
Wet, warm, and in these areas the virus can persist year-round. And so in a dry year there, it tends to be a big outbreak, too.
So when you found that head, what prompted you to save it? Did you recognize that it was a blue top, or it was
My friend found it, and he actually wanted to save it to get a mount. And we just looked at it a little bit and it's like, this is probably a blue tongue case.
yeah.
Because most people, if they accidentally shoot a deer and cannot, or if they shoot a deer and they cannot find it based off a blood trail,
Yeah.
they would have a different coat color, because that's going to be usually
In
October, November. Tomorrow is the opening for bow hunting, and it would be the earliest, and no deer has velvet and a red coat by now. They're all in a darker coat.
Put this down. I want to take a picture of you. This is going to be awesome for the bounty
And then just hold it, pretend you're talking. Oh boy.
No, that was perfect.
Right here.
And I'm going to avoid the note about the restrooms there that's on the board. Hold it up.
Come on, stay.
No, hold it the way you were when you were talking. This is up.
Right
So like pick it up.
Yeah.
Pick it up.
Put it like your head.
Pick it up.
No. Hold it so. Don't get that dirty.
Dr. E's like a dad.
Any other questions?
That's good.
This
Other
is
questions?
really good.
Thanks, Ben.
Where do you keep that? In the garage?
Yeah, a garage.
A garage.
Stays in a garage. Ooh.
That was really good.
All right. Okay. Does anybody have any questions from last time? Last time we talked a little bit about, we introduced catabolism. We talked just briefly about glycolysis. We hardly said anything about it because you guys have learned that before, and you're going to look in the book on page 236. There's dead deer up here. To know the products of glycolysis, and you already know them, right? The Entner-Duderoff pathway we compared to glycolysis and talked about what advantages and disadvantages and what are the differences and what organisms might have it. We talked about the Krebs cycle, which you guys have also heard of before. So this was just in the context of catabolism. Acetyl-CoA enters the acetyl-CoA came from all kinds of places, but in the context of what we were talking about last time, it was coming from, where did that baby go? Right here. It was coming from pyruvate that was derived from the Embden-Meyerhoff pathway. Oh, my pens are still in my pocket. Right here. So this pyruvate, and we'll talk later today about many places the pyruvate can come from. It doesn't just come from glycolysis, and not every organism even uses the Embden-Meyerhoff pathway. But in this context, it came from there. It gets converted to acetyl-CoA, which goes in both. There's 2 carbons. This is a 2-carbon molecule. And that gets oxidized, and the electrons come out. You get a little bit of ATP or GTP, which is relatively minor, we said, right? There's also ATP. And then at the end, you get oxaloacetate. And the big news in terms of energy is this, the electrons that you get if you have the ability to use the electron transport chain. So we said electrons from the Krebs cycle are available for electron transport and respiration. So that brought us to this stuff where we talked about, this is an example of what you've learned before in mitochondria. Here's an example. What was the interesting thing about this figure? This stuff you've seen many times before. These might be different enzymes in here, but this was showing how other things can serve as a source of electrons, not just NADH, but in this case methanol was serving as a source of electrons. And then we talked about, what we didn't talk about, maybe, was other things that these electrons can be used for, or I should say that this energy can be used for. So this shows it being used to make ATP. What other things can the proton motive force that's generated be used for? So electrons for electron transport chain. generally to make a proton gradient. Or proton motive force, right? So proton motive force, we talked about that a few days ago, and we said that was an energy source for something in addition to making ATP. What was the other thing it can do?
Motor the flagella?
Yep, motor the flagellum. That's the energy source for motility. That's shown on here. Bacterial flagellar rotation. Also, when you guys were in cell biology, you learned about active transport and how the proton motive force sometimes is used for symport, and you have protons building up outside there, and that helps bring molecules across the membrane. It also generates electron donors that are used for other kinds of reactions. So far, because we're talking about catabolism, we've talked about NADH as being something that just goes over and gives the electrons to the electron transport chain, but of course it can be used for other kinds of reactions as well. So we're not going to talk about all of the energy balance of this because there's other things I want to talk about. And furthermore, in real life, you don't really have the luxury of taking every single electron from glucose and making it into NADH and using it to generate ATP, right? Part of that's just because of the things we were talking about. If you take one molecule of glucose and oxidize it entirely, you generate proton motive force, but it's not, all of it isn't going to be going to this in real life because you're going to be using it for things like rotate the flagella, you're using the proton motive force for other things. And furthermore, what would be another reason why you wouldn't get to use all of the carbon that comes through here and oxidize it all and make it and get all the electrons out and use it to make NADH? What else would be going on in the real world?
Those are intermediates for different pathways.
They're intermediates for different pathways. So imagine if you're growing E. coli. So if you grow E. coli in nothing but glucose, well, it wouldn't grow, because of course it has to have nitrogen. But imagine you gave it like ammonium chloride for nitrogen and all the other trace elements it needs. It could grow with nothing but glucose, but it would have to take the glucose and take some of the substrate out of there at this level to make its nucleotides, and it would be taking some of the substrates out of here to make its amino acids, and every other carbon-containing molecule in the cell would have to be made from this carbon. So it can't reduce every single electron out of, it can't oxidize every single electron out of there to make NADH because it's using it for other stuff. So this is the kind of thing that's helpful if you've never heard of this before, because you realize that there's energy, but it's not something that's going to happen in the actual world where the bacteria live. But one thing to take from it is, if you're thinking about these two working together, either glycolysis with the Embden-Meyerhoff pathway or the Dentner-Duderoff pathway, and then the Krebs cycle, what you can take from this is that the energy coming out of here in the form of NADH is way more valuable than an ATP, right? Because you're taking these 6 NADHs from 2 turns of the Krebs cycle and getting 15 ATP out of it if you have the ability to do oxidative phosphorylation, which is another name for what? What's oxidative phosphorylation? That's just respiration, right? As opposed to substrate level phosphorylation, which is right here. So did you learn the difference between oxidative phosphorylation and substrate level phosphorylation? Substrate level is when you have the phosphate coming from an organic carrier, like up here. Here's substrate level phosphorylation. 1,3-bisphosphoglycerate gives up a phosphate, and ADP is converted to ATP. That's substrate level phosphorylation, as opposed to this over here, which is oxidative phosphorylation. Okay, so we talked about respiration. We talked about anaerobic respiration, which is when you're using an electron transport chain with something other than oxygen at the end. So would that mean that fermentation would be anaerobic respiration? Anyone? I know you know. I know you know too. What, Ben?
Not necessarily. It wouldn't necessarily be anaerobic. Like,
I
well, I mean,
think you might be answering a different question. The question is, would fermentation be considered anaerobic respiration? What were you going to say, Tony? Right. Right, so if you're doing anaerobic respiration, you're not using fermentation, because fermentation uses an electron acceptor. So you were on the right track using an electron acceptor, but the electron acceptor for fermentation is endogenous. It's within part of the pathway and part of what the cells are using. Whereas the anaerobic respiration is using some kind of an outside electron acceptor. So let's talk about fermentation. So fermentation, there are a few themes about fermentation. You guys learned about this. And epizootic means that humans cannot get it, right?
Yes.
Okay, good, because I just got it all over me. That's okay. It's not the dirtiest thing I've done. Fermentation. So you guys have learned about fermentation before in the context of using sugars. We're going to talk about some other places that fermentation is used. But generally, it's associated with the regeneration of NAD+. So the simple answer to why does fermentation occur is that it allows NAD+ to be regenerated so that oxidation can continue. Assuming that is that NAD+ is what's receiving the electrons from the substrates that are oxidized. So there are four general themes in there that go with fermentation. One is that NADH is oxidized to NAD+. Another one is a generality, not always exactly true, and that is that the electron acceptor is pyruvate or a pyruvate derivative. I'm going to say is often pyruvate. Whoops, pyruvate. Or crap. Pyruvate. Or it's often pyruvate or pyruvate derivative, which you can totally remember from learning about this before in the context of using glucose as an electron source. Other things that happen, or that are associated with it, is that oxygen is not needed. But that doesn't mean it can't happen in the presence of oxygen. It's just that certain certain enzymes involved are oxygen sensitive, but there are kinds of fermentation that can happen. The other thing is what Tony was saying, there's no electron transport chain. So there's no electron transport chain. Does that mean that the cell doesn't need a proton motive force? You still have to have a proton motive force. Why is that?
To move stuff, and then also you need protons in the cell, like constantly, just for like a gradient.
They always have to have a gradient because they're moving stuff, as you said. They still have to have an energized membrane, so they have to have other ways to generate the proton motive force. What would that be? So if this is your membrane, you still have to have protons out here. But there's no proton motive force. Did you ever learn how that happens? It's a big waste. It's the reverse of the ATPase. So they have to use ATP, it costs them ATP, to move protons out here. So not only is there less energy generated, they have to use their energy doing things that ordinarily you wouldn't have to expect. So let's look at some examples of fermentation. Here's kind of an overview of fermentation pathways. And this is not exclusive, but what it's showing, the point of this picture is that if you're using glycolysis and you make glyceraldehyde 3-phosphate and you use NAD+ to be reduced and receive the electrons that are leaving the substrate, you generate NADH. And if that's the only energy you can make up here, it doesn't show that there's ATP made, but there is, you have to regenerate the NAD+ so that this reaction can continue. And so the electrons on the NADH get wasted. They get used to reduce some other thing, like pyruvate, which is shown here, to make lactate. Or here's another example where pyruvate's converted into something else, and then that, whatever X is, gets reduced to make NAD+, which can then allow this to continue, so that they can get ATP out of there. So let's look at, so why do we care about fermentation? Bacteria have to ferment. We're going to talk about some more examples of fermentation reactions that can occur. But in terms of human use, keep you guys on board, why is there any kind of human use for these products? Or what would be some examples of products that we can use? Brandon?
Ethanol?
Ethanol, bread. So so ethanol used for fuel, for beverages. Tony, what are the products that are used in bread? Are you the one who said bread? The products are CO2, right? That's the leavening for bread. It says leavening for bread. Carbonation. Can you think of other foods that involve fermentation? Cheese, which would be, what does fermentation give cheese? It's the organic acids. And CO2. If you have Swiss cheese, the holes in Swiss cheese are due to the fermentation products of the microbes that live in there. They make CO2, and it makes bubbles in the cheese when the cheese is still soft, and all those holes in there are the result of those bubbles. So this is a figure in your book that shows many of this is not an exhaustive list of all the possible fermentation products, but it shows some pathways. These, of course, are all enzymes in blue, and down here at the bottom it shows some microbes that are associated with them. So this one right here, pyruvate to ethanol, you've heard about before. That's what yeast do, and some kinds of bacteria do it. Pyruvate to lactate, that's a super common reaction that is catalyzed by a whole bunch of different kinds of bacteria, including those that are responsible for making pickles and sauerkraut and kimchi. So that would be under this category of organic acids like lactate, lactic acid. So there's all kinds of foods that involve fermentation steps, and the flavors of the food come from a lot of these different kinds of products. Sometimes the fermentation results in things that you wouldn't associate with food at all, like organic solvents. These reactions in number 7 are things that Clostridia do. Clostridia we're going to talk about how they ferment amino acids in a little bit. They use pyruvate or pyruvate derivative as an electron acceptor, but they don't start with sugar, they start with amino acids, and we're going to talk about that soon. So I wanted to show you some fermentation. Hold on here. This is a figure that I believe Dan Schmidtman found for us. It shows about secondary metabolites that are produced by lactobacillus. You don't have to memorize all of these, but the point that I want to make is that there's all kinds of fermentation products that aren't mentioned in the book that can be made as secondary metabolites, along with the lactic acid that these guys make. So these are all things that you can think of as either flavorings or odors that you can detect in different kinds of fermented food. For example, formate mild sour at high concentrations. Propionate body odor, that's like the dirty gym sock, dirty foot smell. Also, if you get that really cheap Parmesan cheese at the Pizza Hut, that dirty, that dusty kind of Parmesan cheese, and smell it, it's got that vomity smell, that's propionic acid or propionate. You'll never be able to eat it again. Ethyl propionate, so there's different versions of this. Pineapple and kiwi. Some of them are more like solventy smells. These are actually solvents, like ethyl acetate, hexane, heptane, octane. Those are all things that can be made by bacteria when they ferment. Things that are generally made in relatively small amounts, but they can be important for these kinds of pathways. Another thing that's going to be important about these is that particular organisms make characteristic compounds when they ferment, and that can be used to identify the microbes. So this can be used to identify microbes. So when are we going to do unknowns? Like around Thanksgiving? Probably Thanksgiving-ish. You guys, what? Ish. I can't remember exactly. But you guys are going to be doing bacterial unknowns in lab, which is really fun. And one of the ways that you can distinguish between certain groups is based on how low they make the pH when they ferment. How much acid they make, and whether they make mostly acid. And so this is one of the ways you can get an idea about what microorganisms are present in a sample. So I want you guys to know some examples of fermentation products from microorganisms and what they're used for. So these are just some of the examples. This table is good. This is figure 1120. So they're used for making food. They're used for making fuel. They're used for making solvents. There are fermentation steps in the processing of cocoa beans for making chocolate, and in making processing coffee beans. Before you can get the shell, the fruit off of the coffee bean, there's a fermentation process that it goes through. I heard that there's a really expensive kind of coffee that is made from coffee beans that have been ingested by a particular animal called the civet cat. And so it eats these beans and it goes through the cat. It's not a cat really, it's a civet cat, which is sort of like a mongoose. And the bacteria in the guts of the mongoose digest the things, and then it craps out the coffee beans, and you collect them and grind them up and make them into coffee. Ben?
On that arrow, on the chart, where you list all the solvents, one of the steps is acetyl-CoA. So at that point, why would they not go into the Krebs cycle?
That's a good question. So some organisms don't have the whole entire Krebs cycle. They just have portions of the Krebs cycle. And the other thing is, if this went into the Krebs cycle and it generated NADH, that would add to the problem that they have, right? Because what they really need is NAD+. So if they had the Krebs cycle, then they could do something way more efficient. Or if they had a respiratory chain that they could pass the electrons to. So basically, and that brings up a good point. Why would an organism do this and be so wasteful as to use pyruvate as an electron acceptor when pyruvate is full of electrons still? So why would that be? Why would an organism ferment when you... So does everybody get why it's wasteful? Somebody tell me why it's wasteful, Luke.
Just because you have a high energy molecule that you could use to make a ton of ATP, but instead it's going out some different pathway.
That's right. It's a high molecule, a high energy molecule you could use to get more energy, but instead it's being used as a sink for electrons. And you're not able to get the energy out. So why would an organism do that? Yep, the environment doesn't allow for it, so that would mean that they were organisms that didn't have an electron acceptor, right? So they either ran out of oxygen and now they're screwed, and so they have to do something else. Or they never did have oxygen and they're not anaerobic. So there's some organisms that are respiring organisms but don't use oxygen. They're strictly anaerobic respiring organisms. So they don't have a terminal electron acceptor, or sometimes it's because they don't have the electron transport chain. They don't have the genes required to make the enzymes to put all that stuff in the membrane. So it's easy to forget, potentially, that all of these blue things and all of these arrows are the products of genes, and not every organism has all of those genes. Or they're the product of enzymes. Sometimes they are something that gets made by the enzymes. So the organisms are doing this because they don't have a choice. So what happens sometimes is that in the real world, not in a test tube, but in the real world, these organisms are living next to some other organism that consumes the end product. So for example, acetate and ethanol, like you said, why would this is so wasteful? Some other organism, if this were happening out in the environment, some other organism would be eating this stuff up, making it more favorable. So it's using this up and keeping the energetics such that they have an easy time doing this. And then that serves as a food source for something else. Okay?
So just to clarify, anaerobic doesn't mean no electron transport chain. It just means that they're not using oxygen as their final electron acceptor. So
Anaerobic,
you can still have an electron transport chain, but use something else as your
yes,
final acceptor.
that's right. And we talked a little bit about that the other day. These are some examples of things that organisms could use. So nitrate could be used as an electron acceptor and generate nitrite. It can be used to generate a bunch of other things. Sulfate to make sulfide, CO2 to make methane. CO2 can be used to make acetate, depending on what organism it is. All of these inorganic things can be used as terminal electron acceptors and reduced, and then make these products. Christian?
So respiration in a sense is basically referring to your ability to do oxidative phosphorylation.
Yep, regardless of what the term, so that's true. But don't anybody think that oxidative phosphorylation requires oxygen? It's not, that's not why the word ox is in there, right? It's oxidative because something is being oxidized. So you can do oxidative phosphorylation with terminal electron acceptors other than oxygen. Everybody good? Okay, so I want to show you, so we were saying before that not only can microbes do this with sugar, there's some microbes that don't use sugar at all. So this is one of those times where we're turning to a different page in the book. Don't everybody panic. We're still talking about fermentation. This is still about catabolism, but we've gone to chapter 23. Did everybody hear that? This is figure 23.4, and we're still talking about catabolism. This is a reaction known as the Stickland reaction. Let me write that down. Stickland reaction. This is used for fermentation of amino acids. So there's certain microbes that live in soil and microbes that live in your mouth, microbes that live all over the place that, in order to compete, don't use sugar, because sugar can be taken up by so many different things. They have the ability to use amino acids as their source of energy. And in this reaction called the Stickland reaction, it requires two amino acids, and in this example it's alanine and glycine. And alanine, right here, gives up electrons, it becomes oxidized. So remember, this is where the energy is coming from, in this case, one of the places, to make NADH, and that gets converted into pyruvate. The NADH, remember, for fermentation has to be regenerated so the whole thing can happen, and it gets regenerated by reducing glycine up here. So the glycine gets reduced, and in the process of that, it gets deaminated. Where's the stylus? Gets deaminated. Oh crap, I'm not ready for that. Huh. Just gets worse and worse. Gets deaminated and you get two acetates out of it. So over here you have pyruvate. The pyruvate continues. Remember we said before that pyruvate still has energy in it. And so this is a case where the pyruvate gets reduced a little bit more. Or the pyruvate gets oxidized, I'm sorry. It gives up electrons and gets oxidized. Generates NADH, which is taken care of again here. Both of these are used over here. The pyruvate gets converted into acetyl-CoA, and here you end up with substrate level phosphorylation. So acetyl phosphate serves as a source of phosphate for substrate level phosphorylation for production of ATP. So this is a case where fermentation is occurring without using sugar. So let's see if all the rules still apply. What were the four different things we said were characteristics of fermentation? One is that the NADH is getting oxidized so that the reactions can continue. That's happening right here. Oxygen's not needed. That was one of them, right? One was that you don't have an electron transport chain. So if you were able to respire, you could oxidize the NADH using electron transport chain to make NAD+, but you can't do that because you don't have one. And what was the third generality about fermentation?
The electron acceptor is often pyruvate or pyruvate.
Yep, this is the pyruvate. So this is similar. This is glycine. You can make amino acids into pyruvate pretty easily by taking off the R group and taking off the amino group. So this would be sort of your pyruvate derivative, or it would be the electron acceptor, which is pretty similar. So where would this happen? A case where this happens a lot is with Clostridia. So Clostridium tetani is an organism that lives in the soil. If it gets into your skin or punctured into your tissues, the spores can germinate. Remember we talked about spore forming and this being an example of a spore former. If they germinate into vegetative cells in your tissues, they can chew up your tissues, which is really great. But before they do that, they would kill you with a toxin they make. But say it's really the Clostridium in the dead deer. They're not going to kill the dead deer because it's dead, but what they would do is export exoproteases and chop up the deer tissue with the exoproteases, and then take in the amino acids and the peptides right here. In this case, they have a whole bunch of sodium ion transporters, which you don't have to memorize for this, but it's a little bit interesting here in a second. They bring in the amino acids, they convert them down into pyruvate, and there's fermentation reactions going on in there. And then in some cases you can see, remember somebody just a second ago asked about, Ben asked about acetyl-CoA, can't they use the Krebs cycle? They don't have the whole entire Krebs cycle, but they do have some enzymes of it, like here's fumarate being converted to succinate. They have some of the enzymes that they need in order to generate some of the intermediates that are useful. But they're making a living by taking up all kinds of amino acids and making energy from them using the Stickland reaction, making pyruvate, which they can use as an electron acceptor. And there's all kinds of things they also make from that, like lactic acid. Why do you suppose that they have, what would be an advantage for these guys to have a sodium-driven transporter rather than a proton-driven transporter? Everybody think about that for a second. So, maybe you can use a potassium. I wasn't thinking about potassium. What were you going to say, Ben?
I was gonna say they're probably in a high sodium environment if they're in dead tissue. I feel like that would probably be high sodium, so they wouldn't necessarily have to put as much work in to building up that.
Does they already have sodium out there? That could be. I don't know how much sodium is in tissue. So there's two things I don't know today. I don't know what ungulates are. I did learn it. I'm a microbiologist, not an animal guy. I don't know. I don't know how much sodium is out there in dead animals. What I was thinking about is that these organisms, because they don't have an electron transport chain and they're not respiring, they're not building a proton gradient. So remember when we talked about proton motive force and we said the protons are used for the flagella and for the ATPase and for transporting stuff? These guys aren't doing that. They aren't, they have precious little protons out there, and for them, it makes sense to have a different system. Let's see here. What else do we want to say about fermentation today? How far behind are we? So today's the 22nd. So we're going to talk next time briefly about the beautiful topic of chemolithoautotrophy. So if you guys want to read ahead, I don't think I can do it in 4 minutes, but just a second here. What is chemolithoautotrophy? So what have we been talking about today? Which category of, do you remember all your potential categories? Hold on, let's look at that. We're not going to waste these 4 minutes. choices up here. Clearly it's not photolithoautotrophy. Where have you guys, so you guys are going to get to see photolithoautotrophy and photoorganoheterotrophs in your Winogradsky column. Some of them already have some. Chemolithoautotrophy, chemolithoheterotrophy, chemoorganoheterotrophy. Those are the three categories that we've been talking, one of the three we've been talking about. Which one have we been talking about lately? Alex, did you say that? Chemoorganoheterotrophy, which means what? They're using chemicals to get their energy, and they're using chemicals to get their electrons, and they're using fixed organic material as opposed to CO2. So what we're going to talk about next time is chemolithoautotrophy, in which they're getting their electrons from inorganic material. They're getting their carbon from CO2, and they've got some special constraints. So imagine if you have, yeah, I know better to start this now. Anybody have any questions? We're good. All right, we're going to talk about chemolithoautotrophy on Monday.